My name is Gabriel Cardenas, and I'll be speaking today about the Warburg Effect in cancer metabolism. So, in the early 20th century, a German physiologist named Otto Heinrich Warburg observed that cancer cells had higher rates of glycolysis, despite the availability of adequate oxygen levels. The aerobic glycolysis is also known as the Warburg Effect. The Warburg Effect can be seen in two different aspects, and that is the cancer metabolism in plants and photosynthesis. I will be speaking about cancer and metabolism today. So it is a major metabolic shift, reprogramming energy metabolism. Most malignant cells get their energy from anaerobic metabolism at rates of glycolysis of up to 200-fold greater than oxidative metabolism of pyruvate and mitochondria. The reliance of most cancer cells on glycolysis rather than oxidation to meet their metabolic needs. So every cell needs their energy, and we can get energy through the mitochondria, which yields about 36 ATP through the oxygen-dependent process of oxidative phosphorylation, or cytoplasm, which yields about 2 ATP that doesn't require oxygen through glycolysis. So in this picture, we see how a cell gets energy regularly through a normal cell. In the next picture, we see a cancer cell, and we'll just relate these two pictures. So oxidative phosphorylation and ATP are significantly reduced. Glucose uptake is increased. So the increase of GLUT1 receptor facilitates transfer of glucose across the cell membrane. The increased glycolysis means that there is an increase of pyruvate. So in this cancer cell, the ATP generated is from the glycolysis. There is also an increased conversion of pyruvate to lactate, in which lactic acid causes a decrease in pH. This all happens despite oxygen, known as a Warburg effect. So cancer cell, they manage to activate glycolysis despite the presence of oxygen by the overexpression of hypoxia-inducible factor 1, known as HIF1, which increases the transcription of many genes which encodes proteins favoring cancer such as angiogens, apoptosis resistance, and glucose metabolism. HIF1 increases the glucose uptake by upregulating GLUT1 expression. HIF1 increases glucose phosphorylation by upregulating hexokinase 2 expression. The increase of HIF1 has similar effect as a loss of p53 function. So why do we care? So this effect led Warburg to assume that the fundamental change in cancer was a metabolic rather than genetic. This is not a cancer specific, it is evident in rapidly growing normal tissues too. So the property of high uptake of glucose is used in PET scanning of solid tumors. Numerous glycolytic inhibitors have been developed which can be potential anti-cancer drugs. Warburg effect is a growth-promoting metabolic alteration in cancer cell. And these are my citations. Thank you.